Okay,
Yes.
are we all set?
I think so.
Excellent. So I think you can hear me without microphone, and I hope that those on Zoom can also hear me. So I'd like to welcome all of us, and also especially Shreya's parents for today's defense of Shreya Palchaudhuri. And so Shreya started her work in my lab quite a lot of years ago, 6 years ago. And when she started, I thought, oh, this is going to be a great PhD. And it turned out it was a great PhD. But in between, there were a few problems. There was actually a retracted paper that was naturally not due to any of Shreya's fault, but I wanted to mention it here. And there was also a pandemic in between of 2 years that kept us all busy a bit longer. But today now, Shreya's big day has come, and I'm really happy to be here and that we are all here. And I'm looking much forward to Shreya's public defense. Thanks a lot.
So, thank you, Ralph, for the introduction and everyone for being here today and sharing this special occasion with me. But let's dive right into science. And thanks also to people on Zoom. Lots of people I see on Zoom today. So my project, as Ralph said, began a long while ago, and it's titled An Insular Cortex to Lateral Amygdala Network in Fear Learning. And I'll try to explain a bit of what I was doing these few years, because a lot of you here sort of know what I was doing, but maybe not everyone. So yes, I did my PhD in the lab of Ralph Schneggenberger at the Laboratory of Synaptic Mechanisms. So why do we essentially need the brain? Um, Amongst a lot of other functions, we can think that the brain is needed in order for us to perceive threatening stimuli in the environment. We need to perceive threats. Why? Because it is so important in order to survive. Our survival depends on whether we can perceive these threats. And threats are universal, because not only are threats there for a woman in New York, let's say, they could also be lurking in the wild. And this makes this circuitry very important across species, from animals right across to humans. So, but how does the brain get involved in this process? We can think of the brain as our control box, where it sort of accumulates sensory information, and sensory information across all modalities like touch, taste, smell, hearing, and so on. And the brain processes these. And depending on the context in which these sensory stimuli were seen or felt, the brain can then adapt them in order to generate a behavior. And the way that it does this is through neurons. The brain is composed of millions and millions of neurons that are connected to each other through these synaptic connections. And I will talk a bit more about this. So let's take an example. Let's take hearing as an example and think, let's say you go to the disco and you hear your favorite song, and your immediate reaction to this is to go dance, right? But if we hear the same music, the same peppy track, let's say you're going somewhere in a car and you have this music on, but a car hits you from behind, that gets laid down as a very, very aversive memory. So the next time you hear your same favorite song, you might not have the similar reaction as you had to the disco. You either try to avoid it, you try to run away, or you really sit down and it sends chills down your spine. So that's what we call freezing when we come to a mouse model. So how do we study this behavior in the lab and in the mouse model? So we have what is called fear learning. It's a modified form of Pavlovian conditioning, and it's a 3-day paradigm. So on the first day, the mouse is put in a chamber, and a normal, a neutral tone is played. And what we record as an output is this freezing behavior that I told you about. So the mice basically, if they're afraid, they sit in a corner and they freeze. But if you play a normal tone that the mouse has never heard before, it really never freezes. You see it very flat here. Now, on the next day, you play the same tone in the same chamber, but every time you play the tone, there's a mild foot shock that's followed. So, tone followed by the foot shock, which you see here in blue, and then followed by red. I'm explaining this because this plot will come up a bit later. And now the mouse becomes more and more afraid of the tone as time goes by, because the moment it hears the tone a little later, it feels, oh my God, the footchuck is going to arrive. So this is what we call the training day. But if you look at day 3 or the next day, it's a completely different chamber that the mouse is placed in. It's a different smell. The floor has a different texture, different lights. Now, if you place the mouse and play the same tone that was played the 2 days before, the mouse freezes very specifically to the presentations of the tone that you see here, which means that what was initially a neutral tone stimulus for the animal has come to mean something aversive for it. It's really specifically afraid of the tone. And this is what we call the conditioned stimulus, and I will use the term CS to refer to it. And the unconditioned stimulus, in this case, is the footchuck, because it is innately aversive for the animal. And this is what is guiding the tone from a neutral meaning to an aversive meaning for the animal. And decades of research has said that the lateral amygdala, a structure in the brain, is known to be the center that receives both footchuck and tone information. So this is the structure where information about both these stimuli converge, and there are plasticity mechanisms, or there are certain cellular mechanisms that happen within the structure that are then laying down this memory. So this is also important because the lateral amygdala sort of can be traced across all sorts of species, right from lizards right up to humans, right? And so again, a survival circuit must be important in order for humans to survive. And this is why it makes sense to study something like this. So let's get down to the science part of it. So where does the lateral amygdala receive its information? A lot of research has shown that the tone information reaches the lateral amygdala through these cortical inputs, so the cortex of the brain, as well as certain other areas like the thalamic areas, all bring in auditory or tone information. And little is known about how the footchuck information reaches the brain. And so this is the question we wanted to ask, is if the lateral amygdala really brings in both sorts of information, where does the footchuck information that comes to the lateral amygdala, where does it come from? So the first thing we need to know is how information flow actually happens through the brain. So let's think of 2 regions, or maybe 3 regions in this case. So region C has a neuron shown here in pink, and this receives information from region A here. And so neurons in region A connect. So this is the cell body of a neuron, and you see dendrites here. And this axon sends its terminals right across to region C, where it makes contacts on the neuron in region C, right? And these synaptic contacts are actually called synapses. What happens is this presynaptic neuron that you see here can activate the postsynaptic neuron if this is an excitatory connection. So when a presynaptic neuron is active, there are molecules released at the synaptic cleft that then bind to some receptors, some molecules on the postsynaptic neuron, cause positive ions to flow through. So this neuron then becomes less negative, so it becomes active, basically. So what can happen is a presynaptic neuron can activate the postsynaptic neuron if it's an excitatory connection. So knowing this, and what we spoke about before, the question we asked was which brain area brings aversive information to the lateral amygdala? And we tried to think a bit broad and first do anatomical experiments. So in this case, what we try to do is we can inject a virus or a dye or some sort of label that's a fluorescent marker. We can inject it into the brain in a certain spot. We can cut sections of the mouse brain so that we see this. And this is what it looks like under the fluorescent microscope. And then we can overlay it with a brain atlas to know where we injected and where these neurons can project to. So we did some anatomical experiments. The first thing I will talk of is the retrograde tracing approach. So in this case, what we did was we injected a cholera toxin B. Don't worry, it's a safe thing to use. So we injected it into region C, let's say. What happens is this is taken up specifically by synaptic terminals in region C. And this travels retrogradely, this is why it's called a retrograde approach, back to the cell bodies. So any cell that projects to this region C will turn purple because it takes up this cholera toxin here. So now we can look across the brain, and wherever we find cells, we know that these neurons project to region C. And this is what we did in the lateral amygdala. So we injected CTB in the LA. Here you see my injection site. And interestingly, we saw cells in an area called the insular cortex, both on the ipsilateral side, so on the same side of the brain as the lateral amygdala here, and on the contralateral side, so also on the contralateral hemisphere. And especially, particularly in the posterior part of the insular cortex, we saw cells in many other areas of the brain, but the insula sort of seemed interesting to us because the insula is a very special area of the cortex which has a role in many different functions. So somatosensation, pain, hearing, even about interoception, so like bodily sensations, as well as taste and gustation. And this became interesting to us because we were looking for an aversive nucleus that would send information to the lateral amygdala. So the posterior insular cortex, because it had a role in nociception or pain signaling, became interesting to us. So this is the area that we are studying. So the insular cortex stretches quite anteroposteriorly in the mouse brain, but we are more specifically located in the posterior part of it. And it's also interesting that literature has shown that the insula itself receives, actually the posterior insula itself, receives heavy information from sensory cortices, which are themselves known to have a role in somatosensory and pain processing. So with all this information in mind, I set down the aims of the thesis to first understand whether this posterior insular cortex to lateral amygdala connection actually exists, because so far we've seen it with anatomy, we need to functionally test this. And then we wanted to see whether the posterior insular cortex itself has a role in fear learning, in auditory cued fear memory formation. And the third aim was to know whether or what sort of information this posterior insula to LA connection actually carries. So to tackle the first aim, I showed you a retrograde experiment, and now I'm showing you an anterograde or forward tracing experiment. So in this case, I injected a specific virus into the posterior insular cortex, which has two components. And I should mention here that this virus was custom made by Alexei in our lab. And so the two components are the synaptophysin mCherry. This is a protein that sticks to the synaptic terminals here, and mCherry is a red fluorescent protein. So it sticks, it makes basically the synaptic terminals turn red. And the GFP label sticks to the cytoplasm of the cell. So you know where you injected in the brain, and you know which area these, if you see red puncta, you know that these green neurons are actually projecting to these areas in the brain. And again, you can look through the mouse brain. And this is exactly what we did. So we had an injection in the insular cortex, and we saw heavy labeling in the lateral amygdala, both of the ipsilateral as well as the contralateral side, amongst other different areas in the brain. So indeed, anatomically, it seems that the insula does project to the LA, but is it really a functional connection? To understand this, we next move to what is called as optogenetics. So this is two gentlemen, Ed Boyden and Carl Deisseroth, revolutionized the field of neuroscience by bringing up optogenetics. So these are proteins that can be expressed on the membrane of neurons to make them excitable or sensitive to light. So what you can do is you can express this protein, channel rhodopsin, and I'm using a variant of this called Chronos. And through a virus injection, you can plant these proteins basically on the membrane of the neurons. And now if you shine light, sodium currents will enter the cell. Sodium ions will enter the cell basically and activate the cell. As opposed to this, there are inhibitory options that I will come to later, which if you shine light on them, they will either cause anions to flow through, thereby silencing the neuron, or they will allow positive ions to flow out, also silencing the neuron, right? So basically, optogenetics allows us to manipulate the activity of neurons in vivo while the mouse is doing behavior, or in other experiments such as ex vivo electrophysiology. So this is what I did. injected a virus into the insular cortex, which had a chronos. Now, if I shine light on the terminals here, and if I if I put in an electrode into the cell, into the neuron, if these two neurons are connected, and if shining light can activate neuron in the insular cortex, I should be able to see currents here because sodium ions are flowing through. And this is what I see. So if I increase light intensity, I see currents of higher and higher sizes. So again, I'm showing you my injection in the insular cortex, and this is where I'm recording in the LA. This is a cell that I'm recording from. And indeed, I see inward currents flowing into the cell with an average amplitude of around 200 picoamps. What I actually saw, interestingly, was that when I injected both insular cortices, so on two sides of the brain, current sizes were double than when I injected unilaterally. So again, going back to the phenomenon that the insular cortex not only projects to the lateral amygdala of one side, but also to the lateral amygdala of the contralateral side. And another cool thing we can test is we can see what the delay is when you shine the light pulse and when you see the current rising. And we saw that it was around 3 milliseconds, which means that it is one synaptic delay. So if it was to go from this neuron to another neuron and back, then it would be a bigger delay, right? If it's one synapse delay, it's around, the shortest is around 3 milliseconds. So this is a functional connection we've seen so far. But does the insula itself have a role in auditory cued fear memory formation? In order to know this, we went back to optogenetics, but this time we're trying to silence the neurons instead of activating them. So the protein we are expressing this time is archirhodopsin or ARCH. So what we do is inject this ARCH into the insular cortex, and now we can shine light to inhibit this neuron and hopefully inhibit its downstream neurons as well. So what we can do is we can implant these tiny optic fibers into the brain of the mouse to guide the light to the specific nucleus, so in this case, the insular cortex. And with the help of wires, then, while the mouse is moving around, we can specifically shut off the neurons during a specific phase of the behavior. So what did we do? So we injected ARCH bilaterally into the insular cortex and placed optic fibers over it. Now we go back to this 3-day paradigm that I spoke of before. So 6 tones on the first day, mouse is not afraid, not freezing. On day 2, when the tone is always paired with the foot shock, when the foot shock arrives, we shine light. Every single time there's a foot shock, we shine light so that the foot shock evoked activity of the insular cortex is shut down at that point, because we hypothesize that the insula brings in aversive information. If that is true, if you shut it down, then mice should not learn as much. So what do we see? So on training day, we see the GFP and the ARCH mice. So in black, GFP mice are control mice where there was no ARCH protein, so they were not silenced essentially. So they learn, both the GFP and the ARCH mice, they pretty much learn, which is a bit surprising knowing that you are silencing hopefully an aversive nucleus. But if you look at day 3, you see that the GFP mice or the control mice freeze very nicely to the tone presentations here, but the ARCH mice freeze significantly less to the tone presentations. So this aversive memory may not have been retained, right? So the foot shock evoked activity of the insular cortex must be required for this fear memory formation. So that's about AIM2 then, but we still don't know what information this connection is carrying. So the posterior insular cortex must be bringing in some information to the lateral amygdala, but what is it? So for this, we moved to a miniature microscopic recording of LA neurons. So we implanted this microscope on the head of the mouse, and this is a very specific microscope designed by Inscopix, which is 2 LEDs, why I'll explain later. And so what we can do, again, use optogenetics to silence. So this is halorhodopsin, another of the inhibitory opsins that I told you about. And what we can do is also record activity of the neurons in the lateral amygdala. So this GCAMP is basically a genetically encoded calcium indicator. So whenever the cell is active, there is the intracellular calcium increases. Because the intracellular calcium is bound to a fluorescent tag, whenever the cell is active, you will see fluorescence. And this is what we are recording through the microscope. So microscope is, the green lens is implanted over the LA to record the activity of the neuron whenever it's active. Now, this green lens can also permit light of another wavelength to flow through. And this is what will silence the incoming axons of the insular cortex. So you can silence these neurons and still record what happened to the activity of the LA neurons. So if insular neurons project to the LA and bring in information, when you shine the pink light, then you should see a suppression of this activity. So you record GCAMP activity when the cell is active, and then you see what happens when you shine light to inhibit these neurons. So this is what it looks like when I look through the microscope. So these are tiny individual neurons in the LA, super fluorescent. And now I'm doing again, the mice are going through this 3-day behavior paradigm, and I'm trying to shine light again around the foot shock in order to see whether LA neurons receives its foot shock activity from the insular cortex. So here are examples of few individual cells, and you can see, so the tone comes and the foot shock concomitant with the light pulse. You see that some cells have a robust foot shock response in the LA. Now, this is the second tone with the foot shock, but no light pulse. And if you compare, you can see some cells here that are inhibited by the light, but have a foot shock response. So they must be cells that receive foot shock information from the insular cortex. If we look at all cells across one mouse, you can see that there are quite a few, maybe 1/3 neurons of the 60 neurons that I recorded from in one mouse that are foot shock responsive. So these red ones here show high activity. And now if you shine light on them, not the most robust US responsive or foot shock responsive cells are affected, but there's a population here that seems to suppress its foot shock activity when the light is switched on, meaning that the lateral amygdala neurons must be receiving at least partially its aversive information from the insular cortex. So overall, my PhD thesis has shown that the posterior insular cortex to the lateral amygdala is a functional monosynaptic connection that's also glutamatergic and excitatory. Now, if you silence posterior insular cortex neurons, mice do not show a fear memory on retrieval day. So the foot shock driven activity of the posterior insular cortex is needed for retrieval. And I've also shown you that if I inhibit axons of the insular cortex over the lateral amygdala, the foot shock response of the LA neurons is at least partly affected or suppressed. So I can say that this thesis has established that the insular cortex lateral amygdala connection is a novel pathway in auditory cued fear memory that is both strengthened with learning and also provides information to the lateral amygdala. So with this, I'd like to thank members of my lab, Ralph especially for being my supervisor for all these years, and a bigger acknowledgement follows later. But I also want to thank members of the jury for providing critical comments and fantastic feedback, and EPFL for having hosted me for all these years. So thank you and everyone for being here today. If anyone has questions, I'm happy to answer.
Okay, so how should we go? Would you like to call for questions, or?
I think we can do questions first and then I can do acknowledgements.
Excellent. Let's go. Questions, please.
I do. I feel a bit embarrassed to ask because I've worked with you for so many years. But just I was wondering, because basically in terms of inhibitory viruses, you have two types. You have the arch and you have the halo. And from what I understand, the haloredoxin you showed, I think it's a chloride, it hyperpolarizes by chloride coming in. whereas the arch is photons going in.
Yes.
So it achieves the same thing, but in AIM-2, you used arch, and in AIM-3, you used hill.
Yes.
And I was wondering why you would.
It's a
Very
fantastic. No, it is a fantastic question, because the thing is, it's thought that in the soma, arch is more effective because it drives protons out of the cell, but it doesn't affect the terminals basically. So it can shut off the neuronal activity, but halorhodopsin is. Yes, and halorhodopsin is more specific when you're silencing terminals, because again, the same thing that like the soma can be unaffected, but you can still shut off neurotransmitter release, hopefully. Yes, Julia.
I'm very curious because you showed at the end that there seem to be two populations, some that are affected and some that aren't. So what's your hypothesis about that?
So the reason why it's not affecting all the neurons can be many. The first obvious one is that LA neurons receive footchuck informations from other areas of the brain. This is for probably true. The other thing is that with halorhodopsin, we might not be effectively expressing it in all neurons. Even if we do, we might not be recruiting all of those neurons when we shine light. So all these technical aspects. And the last possibility is that the footchuck itself elicits such a strong US response that simply with halorhodopsin, we cannot silence the terminals enough to block depolarization, basically. But very good question.
Many more, but it's really
You can ask. Yes, Anastasia.
I was interestingly surprised that when you included the insular cortex and then you look into how mice learn the condition behavior, that during exposure, like during day zero, there is no difference, but only on retrieval you see the difference. What is your hypothesis?
Yeah.
Not needed for learning, just for retrieval, or?
So this is something we also need to tease apart a bit more. But for the humans, it's known that the insula doesn't affect the perception of pain. Also, when we look at the mice videos, we can see that the mice really jump to the footchuck. So it's not the perception of the pain that's affected, because this might be more of a direct network. But what the insula is doing is probably playing a role in laying down the memory. So that's what we would think.
So the information about footchuck is not coming directly from the insular cortex.
I think to the lateral amygdala, it must be coming directly. Whatever footchuck information is coming from the insula is directly to the lateral amygdala. But for the perception or for the bodily reaction to it, there might be parallel pathways that are also active. Yes, I still.
And where are the outputs of these neurons? So one possibility is central amygdala. Or
Yes.
they're also directly to the motor area. And if there are pathways via central amygdala, why it's necessary? What kind of effect that would give?
So yes, definitely DLA projects to the central amygdala, but it also can project to the BA and then to the central amygdala. But there must be a finer network of excitation inhibition balance that's needed. We haven't studied the pathways downstream, I have to say. But so you're saying, why is it needed? Why the insula to the LA pathway is needed? Or why? I mean, the central amygdala is sort of the output station of the lateral amygdala, and that is the one that has direct connections to motor output centers that bring about these behavioral effects. So I guess it has to go through the central amygdala. This is my feeling. But it could also have direct downstream connections to motor areas that I don't know of. Yes, Mukul.
Maybe this is very naive, but I was wondering, for how long can you keep the neuron active or inactive? And you have to do it for every footchoke, or you just do it once and then it
So
stays
yeah,
inactive?
you're stepping on a very painful ground here. So for different opsins, they have different time courses. So what I showed you for Chronos, It's a very fast active and fast switching off opsin. So you can shine light for what I did was, in this case, I was doing 1 millisecond light pulses to record currents. But for Chronos, it really follows very well. For other excitatory opsins that we are slowly moving towards in the lab, you can switch it on and it remains active for a long, long time. Sorry, inhibitory opsins, not excitatory opsins. And for the halorhodopsin that I was using, you can keep switching on and off, but within 50 milliseconds, the efficacy decreases to half, basically. So that's a bit the problem of using this.
And the duration of your stimulation is?
So in this case, I was using 3 seconds, yes. So here you can see that it was a 1 second foot shock and a 3 second light pulse. The reason we used this was because we wanted to make sure that it was really switching on before the foot shock arrived. But in hindsight, we could have done the experiment better. Yes.
Question from Sritama.
Yes. Could you read it out?
So it should be
Okay. Hey, Shritama, you can probably talk.
Hi, Sriya. Amazing presentation. Great.
you.
I have one question. So did you actually try to look at like inhibit the neurons during retrieval and see its effect, not during the training, but just while you are retrieving, if you have inhibited them, and did you try to look at it?
You have to come back to a PhD defense 6 months after mine. I think Dennis is going to present this data.
Okay,
in
great.
future.
Thank you.
If I,
Yes, sure.
is the same network active for other kinds of inputs like visual or touch?
We haven't tested. The insula is involved. I don't know about vision, but definitely is involved in somatosensation, is involved in tone processing. This is something we know about in the lab a bit, but we don't know about other inputs. But there have been studies that show that it's involved in conditioned taste aversion. So in taste, definitely. But this is more the posterior insula to central amygdala pathways. We don't know about the LA so much. Yes, Simon.
Completely unrelated to the biology of it, but how do you make sure that, so you're recording quite small currents, right?
Yes.
How do you make sure that you're switching on the light or the foot shock doesn't interfere with your signal?
You mean in the ex vivo electrophysiology in the data before? Or this data? Do you mean this data? Yeah. So we have control experiments to see whether when you switch on the light, no matter in the background, if there is an elevation of the calcium signal. And we've tried this in a different setup with fibrophotometry where it does. But I think the Inscopix microscope, in our hands at least, we've seen that these two wavelengths don't interact as much. So it does seem quite clean. And actually, in order to mention this, we had another set of control experiments in this experiment where instead of halo, we had a PBS injection just to check whether light itself has an effect. And this, even though there was a trend, I have to say, it didn't actually affect neuronal activity. More questions?
Another naive one. So how often do you see contralateral connections in neuroscience in general? Is it very common in different regions?
In cortical connections, yes. From the cortex, yeah, you can expect that this might occur.
Excellent. I think if there are no further questions, let's go to the
Yes, please. So I don't need to share my screen again, right?
Hopefully.
Hopefully.
You never know.
Do you see? Okay, great. But I will. Great. Okay, so yeah, this has been almost a 6-year PhD. sort of what I expected from a neuroscience PhD, I have to say, knowing so many neuroscientists coming and going before me. So I want to thank some people who have contributed immensely to my journey at TPFL in Lausanne, in Switzerland, and through the PhD. So I have to start obviously with Rolf, who was my supervisor. He's definitely the boss, whether it's on sea, land, mountains. You can see him here, he's in his element. I thought I was picky till I moved to the Schneggenburger lab. He is so picky, but he is so right all the time. It's very difficult. When he comes and says there's one typo in your figure, you know, the font size is slightly different. You're like, no, I made sure it was correct. And then you go back and say, no, he was right. So that makes it difficult, but also it's a great learning experience. And thank you, Ralph, for all the support. It's not been an easy PhD, as you know, but he's been the pillar of strength through this 6-year journey, I have to say. I also have to thank members of the jury, first for accepting to be members of the jury and for coming up with fantastic feedback and great questions and really taking the time to go through my thesis in immense detail. Next, I want to move to the administrative assistants who've played such a big role in getting paperwork done and everything on time, and Evgenia for really championing my cause sometimes, knowing how paperwork can be really difficult. And Alice for taking care of all of us PhDs through our careers. I also have to thank my previous mentor, Professor Upinder Bhalla. I learned a lot from him, and he was a great academic influence at a time when really my faith in science seemed to have been shaken. And you can see the similarity between the cake we made for him on his birthday, the eyebrows and the mustache that we always made fun of. So yeah, he will always remain as one of the greatest influences in life. And coming to the ELSM crew, the best lab I could ever have been part of. You see the fun girls and the boring boys. What can I say? But that's not true. We've had some wonderful memories together, both drinking and being in liquid, as you can see here, at birthday parties and just random satellite visits, as we all do. It's been really, really a great time together. I want to specifically thank Superman Alexei, as you saw me call him this evening, a guy for all times. Every help that you need, he's always there. Yeah, what can I say? Call the Ask Alexei helpline when you need it. He will be there for sure. Also want to thank the other postdoc in the lab, funny man Michael. He really carried my weight through during the first years of the PhD, as you can see here, but he was also very generous with jokes, both when it was needed and both when it wasn't. But he was always the life of the lab, and he was my neighbor. Enjoyed the years with him. Really, really miss that he left the lab. Coming to the inimitable Heather, always the ray of sunshine at ELSM, always gorgeous, and always such a ray of support. She was the one taking me out on, impromptu dinners or ice cream when I was feeling gloomy. So thank you for that, Heather. Coming to the other ELSM technicians, Tess, who is here today. Thank you so much for coming. She was also part of my first ever snowman, as you can see here with Heather. So very strong memory there. Barbara for being amazing. Jessica, who was always helping us so much with every histology need ever. And then coming to some ex-ELSM fellows, Breeze, the ever cool Breeze, who was such a nice guy and always helping out so much. Anita, obviously the warmest person ELSM could ever have, and also, easing my way into the lab. Aya for game nights and all the fun dinners that we did, and Wei for simply being himself. Like, no one can beat Wei. And then coming to the ELSM PhDs, Aista for really, really big gratitude for all my firsts in Lausanne, my first clubbing, my first hiking, first friends, too many memories. Dennis for being the most annoying PhD student ever. most annoying colleague, most annoying friend, but also the most lovable. He's the person I will miss most in Elzem, I have to say. Yeah, you can see him here trying to be the cool guy Dennis, which he's not, I can tell you. And this is few minutes before he must have burnt his face, I'm sure. He and the sun have a very love and hate relationship. Beishwan, always wonderful being around. She's such a positive influence in the lab. And she's given me a lot of great memories as well. She's so much fun. And when she needs a calculator, you know, she's also having fun as well. And yeah, the future of Elzem, Elena, a very positive influence on the lab. Keep the positivity going. Don't let Dennis take on with his cynicism. And also the cool guys. We hope we gave you a good time, the students in the lab, Milo and Guillaume. I think Guillaume is on Zoom today. So yeah, thanks for bringing youth to the lab, which we are not. We lost due to our PhD. And so yeah, that's about it with Elzem. And now I want to thank a few friends beyond Elzem. First, obviously, the EPFL Girl Gang. This was a serendipitous journey together. I don't know how, like, so many of us came together in such a small time frame, and we had our PhD journeys together. It was fantastic. Ali, Julia, Jane, who am I missing? Kiara. Yeah, the list goes on. So thanks a lot. But special mention, obviously, to Lucy, who has been my support through everything, even unfaithful sunflowers. We spent so much in summer just trying to grow them, and they weren't nice to us. But thanks, Lucy, for everything. Dolan and Mukul, thank you for being part of my life and being the only Indian influence in my life, actually. And funnily enough, also thanks to Anita for bringing them into my life. Had fun times together. It was a great journey together. And coming to the rooftop party house, obviously, I have to thank Edo immensely for not just making a home for us, but also a place of fun for all our friends. We had like such a great time together, especially the COVID years. Vera for being, well, without flatmates, it would have been a terrible time. We had too many dinners, I have to say. And for Vera for being the baby we could pamper all the time. Much love to you. Kostya, who couldn't be here today because he's down with COVID, unfortunately, but also a great person to be around. And we had really great time, sometimes too much fun, but it was a good few years together. And the new inmates of the rooftop party house, Tommy and Alice, for being, again, young, but also so much fun. We also had great dinners together. That was great. And people who've come all the way from Germany, they are, so Titir, who's my friend from Bachelor's and has, thankfully, she's chosen to do her PhD in Europe. So I had one friend that I knew in Europe. So we traveled a lot of countries together and did a lot of things together, actually. So thank you, Titir and Kaushik, and thanks for coming all the way here today. I also want to thank Friends Beyond Borders and friends who, through odd occasions, also become friends. So firstly, Imroz, thank you for being the friend, philosopher, and guide, but I'd really ask you to be more of a friend and a little less of philosophy and maybe a bit more of a guidance, because I mean, coming from your age, it should happen. Some more guidance. But thank you for being who you are. Want to thank friends who made the journey to EPFL possible. Shaborni, who was my flatmate when I was doing my master's. But more than that, she was actually my homemaker because I had no bed to sleep in, no room. And she was generous enough to say, please share my room. Just walk in. And this really helped me take me through my master's, or probably I would have quit and left. Dipayan for being there for quite a few years, and Shankamala for being annoying, but still sticking it through so many years. I want to thank friends from Bachelor's, the presidency gang. Shurja and Sumit for never being there when you need them, but still they are friends, so you have to carry them around, unfortunately. Rumani for being a wonderful person, but sometimes and sometimes not. But really speaking the truth and saying it out loud when it needs to be said out loud. And especially Shritama, the only fellow neuroscientist who remains, actually. Thank you for traveling borders to see me on any occasion possible and being such a pillar of support through everything. Also want to thank friends from school, all the Cynics and Tubes gang, but don't ask me the meaning of this. Ask me after I've had a few drinks. So Shebonti, Shani, Sanjana, Promita, all of them, but especially Aparna. She's been my friend since we were in the 3rd standard. And thank you for sending a present for my PhD all the way to Switzerland. It's been really nice having you as a friend. And yeah, I have no words, really. I also want to say thank you to my extended family, both from my mom's side and from my dad's side. As you know, Indians are the most gregarious sort. They will get together on any occasion possible. Also want to thank people who are no more and passed away during the course of my PhD, my grandparents who are here in the center, and few people that I miss terribly. And if they were here today, would have made this occasion beautiful. And last of all, my brother, who unleashed the world of science upon me. He bought me my first Nature magazine when I was in the 8th standard and said, you should read carefully. You will not understand everything, but you should know that this is life. And so I read Nature magazines cover to cover, but didn't understand a word. Painful. But yeah, really grateful that he really introduced the world of science to me. And obviously, last but definitely not the least, my parents, for being the people they are, for never allowing me to, limit myself. They are the ones, and their outlook has brought me here today. And thank you so much. And everyone for being here today. And Lausanne, a big thank you. A city cannot be more beautiful than this. Thanks a lot.
Shreya. So thanks a lot for this lovely presentation. And so I'd like to present you with this certificate that you brought to me the other day. Congratulations.
Thank you so much.
Any photos?
Yes, please.
So you should turn this around.
This is your one.
Now we're supposed to smile.
Oh, wow.
Thank you so much.
Congratulations, Shreya.
Thank you.
Okay. Where can I see people on Zoom?
Because you're not.
I'm not on Zoom.
You want to see them?
Yes.
But they see you if you stay there. Yeah.
Oh, okay. I see. Yeah.
No.
Well, maybe I
We
should
cannot
stop the sharing.
It depends.
Oh, I forgot.
As per tradition, the tradition in the LZIM lab is to give an academic hat to a new doctor of science, as well as a present, naturally from the lab.
Yes.
So with you, we continue this great tradition. and you would like to put this
Oh
hat
my God.
on your head.
You do?
It's your Shreya's PhD life and journey,
Yes.
all in one picture.
Yes.
I think you will notice some of the things which are similar, some are not.
Yes.
You can think about it. Alexei has always outdid himself, and you will
Yes, of
maybe
course.
play around with it a
Yes.
bit. We told him not to do it because he's not his co-supervisor, but you know, like he likes to torture himself, so it's fine. In any case, allow me to put it on your head. Okay. You, Doctor of Science, as well as a present, naturally from the lab. So with you, we continue this great tradition, and we would like to put this hat. It's your Shreya's PhD life and journey, all in one picture. I think you will notice some of the things which are similar, some are not. You can think about it. Alexei has always outdid himself, and you will maybe play around with it a bit. We told him not to do it because he's not his co-supervisor, but you know, like he likes to torture himself. So it's fine. In any case, allow me to put it on your head. Okay. Yeah. Here's your present, yes. I hold, yes. Yeah, go a bit closer. And we wish you all the best.
Aisa, you need to be here.
Yeah, Aista, you can come also. Come on.
Yeah, Aisa, come on.
Shreya, you want to walk so that the people in Zoom can see
Oh,
the-
yeah.
the
After we
Zoom
take the
account
photo.
back. Yeah.
Mobile phone. I feel like I'm gonna hit someone. Where do I need to be? Ah, okay.
Yeah, I think. Yeah, that's good. Put it down for a minute.
Yes. Yes.
Hands behind the head. Put it down.
Excellent.
Yes. And now we can proceed.
Yes.
Oh.
So we have a small presentation for
Oh,
Shreya.
I see.
Yeah, so that's not yet over.
Not yet over. So I take it out, right?
Yeah. Very fast.
It's just my last paper, sorry. Sorry, whoa. Close your eyes, Shreya. Close your eyes. Close your eyes. Close your eyes. Okay, closed. So from all the friends, we've been also your friends for so much time. It's if you divide our life, it's a big part of it that we spent together looking at the same things, discovering in science as well, which has been an amazing journey to be together with you for all of us. So we all wanted to share moments that we spent with you. So Shreya, you started here. I think that's one of the first photos that we have here at TPFL from you. And then you were so confident, you were so excited, you were so moved about science and discovering new things. And then you became really confident and you said, okay, I'm going fetching, I'm having time of my life, I'm doing experiments. And you were a very keen scientist, behavioralist. You looked at the behavior of the animals and you wanted to understand what's happening in the mice heads. Why do they feel pain? So we also observed you a little bit from behavioralist perspective. Who are you? So you've really enjoyed the PFL. Your friends in different places. You also loved people, and you've been always the spirit and a big laugh inside, outside as well, for our lunches where we cooked and had lunches together, in the parties, in again more parties. So you see the tendency of Shreya, like being very social, involved everywhere, attending things. It's a pleasure to be around her. More parties. And what did you notice today about Shreya and a lot of people who came here? That she brought a way of dressing. So she was always best dressed, and she always carried it through big and small celebrations, and would always have some small detail that made her very exceptional in that way. You see, home too. She's also a supportive person that came for our birthdays, came for the concerts that we had. She was always there for us. So that's very appreciative of her personality. When we were singing, she would also dance, appreciate, react to it. One big thing that happened for Shreya is that she learned skiing here in Switzerland. And she did that with Bijan. So I think you also have to put that on your CV. And when everyone sent me a lot of photos, I received tons of it. I then started putting them together by your behavior patterns. And one big behavior pattern that I observed was the positive reinforcement that you had to get through your days. So that was a reappearing theme in many of the pictures. And it's continued like that, and then it's continued like that. But it's okay, because you also did that with us. So we should not look at you as a special case. But we all enjoy the positive reinforcement in our lives we do together. Also, Christmases, celebrations we had together with us, and also your special friend during the Christmas times. And traveling in different places. Here, attending the exhibition for the images with Lucy, supporting her, and also traveling in different places in Switzerland. Also, something that's very special about Shreya is that she's a very good cook. So she made a lot of dinners for us, and she made to try us different foods from India. And she's an amazing host as well. So when you come to her place, she's always taking care of you, inviting everyone, really feeling welcome. And that's something that hopefully we will continue having in our postdocs as we will go to the same place. And I'm really happy that you enjoyed your time in Lausanne with all of us. And we really enjoyed the time with you as well. And there are some messages from people that are far away like this. Do you have a guess who can be here? No? So we have a message. Well, you better work.
Hey Shreya, many congratulations on your PhD. I'm really happy for you. See you soon on this continent. Bye bye.
And then I have a quiz. So imagine you in the dark room. You cannot see Shreya, but there are many people. How do you distinguish Shreya? One thing is dancing and you feel the vibration of the air. Another thing is her cooking. And third thing, maybe laugh. What do you think? Laughing? I know. So here we go.
Is it a video or a photo? Take a photo. Take a photo. Come on. Don't take a video.
So that was, I think Ralph has a lot of experiences because the wall is very thick in the PFL, but for the laugh, a very thick. For Tria's laugh, a relatively thin. So we tried to reproduce that.
Congratulations, Tria.
But it was really hard because you have a very special laugh, and then people were a bit getting frustrated because it's hard to reproduce yours.
After 10 minutes trying, the conclusion is, in order to laugh, I need your contagious laughter.
And well, and then it worked. So that's Sylvia saying hello to you. What? So lots of laughs from. And I think that's a very good message that with all the images and photos we've seen and experienced with you, that you make everyone smile and everything smile. So that's for us, it's the message that we want to relate to you, how you make us feel. And we wish you all your dreams to come true in the future. So that's from.
And made by Eista and left in my post box.
Oh yeah, that's true. So I think there you should come.
Tria, laugh. Laugh. Start singing. That's the laugh we needed.
I'm embarrassed about my laugh.
So we also wanted to put together a few gifts from you. You can open them as you like. This one first, this one second.
Okay.
And
it's important.
the sequence is important.
Okay.
Big first.
Okay.
And we also made you a traditional photo book. We had a few misadventures with this though. So if you open it, there are no photos.
Okay. I know. Okay, that's perfect.
The photos didn't arrive. So we hope in the next few days we'll get some photos. So you can see it's months. Yeah, our artistic. Yeah.
But some important messages are already there. So you don't have to do all the job by yourself.
Okay. And we would also like to pass that to everybody at the after hour. So everyone can sign like whatever you want.
Whatever you want.
Don't say that. Thank you.
So congratulations, Dr. Shreya.
Thank you, Dr.
You wanna say?
So we can invite. Yes. So maybe everyone to the apero right now.
And if they want to unmute and say.
Yeah, exactly. While people are,
Yeah.
yeah, people on Zoom, thank you for being here. If you want to unmute and say something. Oh, Kostya!
Hi, Shreya.
Hey!
Congratulations.
Where do I say hey? Thank you, thank you.
Yeah, you're looking roughly in the right direction.
Yes, there. Sorry, I didn't hear you.
Yeah, I just wanted to congratulate you. Yeah.
Thank you,
Yeah,
thank
amazing
you.
defense, and yeah, I'll let you talk to other people.
Yes, but you need to be back in Lausanne. Get well soon, but take yourself here.
Thank you.
And thank you, Guillaume. I saw that. Oh, Mikhail is here.
Yeah, he joined. A lot of people, actually, many more than this.
Were there,
Yeah, a lot of people. I kept adding people because the people were joining.
Congratulations, Shriyo.
Thank you.
Congratulations, Shreya.
Thank you. Talk to you and call you later.
No, but I was really wondering who it was. I think it worked.
Yeah.
Excellent.
Perfect.
Good. Priya, I took
Here,
the
sorry,
certificate.
Aparna.
Don't
Oh,
worry,
yes.
yeah?
Thank you. Yes.
But Aparna said, wait, maybe you don't want to.
Oh, hello, Aparna. I see you. I don't see you, but I saw the wave. Thanks for joining. Oh, there you are. Hello. Yeah, I know it's late in India. You can go sleep. You're muted. You're muted. I can't hear you.
It usually worked.
Sorry, I was just celebrating the end of the pandemic, so I forgot to use how to use Zoom. But anyway, yeah, congratulations. I'll talk to you
Thank
later.
you. Yes, absolutely. And thanks for the present. I've been wearing it.
ঠিক আছে ওসব পরে বলবি যা বেরোতে যা কিরকম
Okay.
Yeah, I think we can switch off the Zoom probably. Everyone, I'll obviously call you later.
I am.